Okay, so these notes are on cell cycle control for the IB exam and the AP exam. Okay, so first off, why do cells even need to bother going through cell division? Why make a copy of themselves? And it's true, many times they don't need to do it, but they should be able to when they do need to. So you have to think back to when we talked about the cell theory. Cells can't get too large. Okay, if the volume of the cell grows at a much faster rate than the surface area of a cell, and the volume is what determines the amount of nutrients and oxygen the cell needs and how much waste products that it's making. But the cell membrane, the surface area of the cell, determines the rate at which those items can enter and exit the cell. So when a cell becomes too large, its surface area to volume ratio becomes too small, and it will eventually die off. So in that situation, the cell needs to divide so it can reduce its volume, but increase its surface area to volume ratio. Okay. There are other times too that cells need to divide for growth of a multicellular organism. So we all, as people, started off as a single zygote, but then eventually, most of us are adult humans or almost adult humans. So we had to go from that one cell to, trillions of cells. So you had to do that through cell division. There's also cell division that occurs for wound repair or disease repair. If you get a cut or an injury, or if you've been sick and you say you've had strep throat and streptococcus bacteria have literally been eating away at the cells lining your throat, then you need to replace those cells. Those are all reasons why cells would divide. This is really tightly controlled, okay, by the cell. So in order to go through cell division, there are 3 checkpoints that the cell has to meet, and they are at the G1 phase, the G2 phase, and the M phase. And we call them the G1 checkpoint, G2 checkpoint, and M checkpoint. If the cell does not meet those checkpoints, it doesn't continue on and go through cell division. Okay. Now, in the long ago, billions of years ago, okay, multicellular organisms had not evolved yet. All organisms were single-celled. It took quite a while for cells to evolve cooperativity, to be able to coexist with other cells and to communicate with them and to know that, hey, we're all part of the same organism, like we have the same DNA, let's work together. However, when you have cancer, that's actually an atavism. That's a throwback to this ancestral trait that cells used to have before we evolved multicellularity. So cancer is when your own cells stop responding to the other cells in you, the organism, and they just start growing all on their own, just like a single-celled organism would do, without regard to any other cells near it or any other signals coming from those cells, which is good in an evolutionary sense for that cell because it's copying and passing on its own genes. But for the multicellular organism, this is bad because that cell is no longer doing its job and it's competing for resources with the cells in your body. And most of the time it outcompetes them, which eventually leads to the death of that organism. So we actually do, when we look at cancers nowadays, we do treat them from an evolutionary perspective. So we try to provide environments and medicines that force them to evolve, not to be quite so nasty. And it's considered kind of a managed disease now, rather than you're cured of it. So here is a picture. This is a breast cancer cell. So the breast cancer cell is this larger blue cell in the back. You can see it has kind of a funny shape. These are other body cells. This is a red blood cell here that are being consumed or subsumed by this breast cancer cell. It's now selfish. So in the G1 phase, growth phase, the cell is physically growing larger in size. It's also, doing all of its metabolism during this phase. But the G1 checkpoint determines should the cell go on to the S phase, synthesis phase, and replicate its DNA. Has the cell reached a large enough size that yes, cell division is necessary? So if the cell is large enough, it passes the checkpoint, it will move on into the S phase. If not, it will just kind of exit the cell cycle and hang out in the G0 phase. Your neurons are an example of this. They don't go through cell division. They stay in the G0 phase throughout the majority of your lifetime. Now, that's different from senescence. Senescence is when cells just stop dividing. They're unable to repair their telomeres anymore, and they can't divide anymore. That's aging. So how cells control the cell cycle depends on a combination of factors. So some are internal controls and some are external controls. So examples of internal controls, okay, it's an enzyme regulated metabolic pathway. So there are different protein molecules. Remember, enzymes are mostly proteins, although some can be RNAs. And these protein molecules exist in differing concentrations throughout the cell cycle. And it's these that control the cycle. Okay, so there's MPF is called mitosis promotion factor. When this is present, then that triggers a metabolic pathway that signals mitosis to start. Now, the MPF, you do have to know that one, mitosis promotion factor, is actually a combination of two different molecules. So there's CDK and cyclin. And CDK is a cyclin-dependent kinase. That's where the CD part comes from. Remember ACE, the ending ACE means it's an enzyme, and this is a kinase. So back when we did the case study on Gleevec, we learned about kinases, okay? They're enzymes that phosphorylate other molecules. So they add, the phosphate from ATP, and they kick off this whole metabolic pathway. Okay, the CDK is always present in the cell. It has a constant amount. And that's the enzyme part because it takes a lot of energy to produce. The cell has to go through transcription and translation. But the enzyme can be reused. Remember, enzymes speed up a metabolic reaction, but they're not themselves a participant in the reaction. They're not affected by it, so they don't get used up. So the CDK can be reused. So it's always present in a constant amount. But cyclin is basically, it's a cofactor. It's an on switch for the CDK. So when cyclin is present, it attaches to the CDK, like you can see in the diagram here. Okay, and when the two are together, the CDK is turned on, and at that point we call it MPF, mitosis promotion factor. Cyclin does get used up in the reaction, so it degrades after it's done this reaction. So cyclin changes its concentration, and it's cyclical. You can see it in the graph up here. It is cyclical. So it rises and falls, and it's actually really tightly correlated with MPF activity, which makes sense since you need cyclin to have MPF. Okay, and it's the cyclin that determines, that pushes the cell through the cell cycle. Okay, if there's not cyclin present, then there will not be mitosis taking place. If there is cyclin present, there will be mitosis taking place. We're going to practice this with this today after the notes. Now, there are also some external controls. Remember, I said that we are multicellular organisms. So all the cells of your body are communicating with one another. I tend to say talking, but it's not, with a voice and sound waves. But they are communicating with chemicals back and forth. And one of these is called PDGF, platelet-derived growth factor. So this chemical will stimulate cell division if it triggers receptor proteins on the membranes of cells. So other cells will make this growth factor, export it from that cell, and then it will travel around in the bloodstream or the interstitial fluid until it hits receptor proteins on the cell membrane of another cell. So here's an example of, here's an example of an experiment where scientists extracted the PDGF, and they did a control where they put cells, this is a culture flask, put cells in here without the PDGF and those with, and you could see this one, the growth took off, and this one it did not. Okay, so this is one method of control. So in this case, if a cell in the middle were damaged, the other cells could make the PDGF and then fill in the hole, basically. Now, sometimes, well, there are inhibition methods to also stop cell growth. So if in that case here, this is called density-dependent inhibition, the cells completely fill this container, so there is no space for the PDGF to trigger the receptor proteins. So no cell growth will take place unless some of the cells are removed, and then PDGF can be made, and then cell division will take place in these cells around the hole, and it'll fill in. There's also anchorage dependence. A lot of animal cells for multicellular organisms don't really grow well just in a beaker. If you had a beaker of nutrient broth, they're anchorage dependent. So that's one of the defining characteristics of animal cells, is they make extracellular matrix, and that's where cells anchor onto. Okay, so they have to be attached to something, basically. That's part of why us trying to 3D print organs is a little bit difficult, because it's kind of hard to make that extracellular matrix. So right now we are doing things where we're using other animals or donor organs from people who are deceased, and we wash off the cells, use their extracellular matrix, and then 3D print new cells on top of that for organ transplantation. It's actually really cool, but it's tricky because of that anchorage dependence. But cancer cells don't respond to these inhibitions or signals. Okay, so if you have a cancerous cell in the same situation, it will just keep dividing and dividing and dividing until you get that characteristic like tumor or lump. So cancer results when you have uncontrolled cell division. Okay, and there are different stages of cancers, and some cancers are worse than others, depending on how uncontrolled it is. So first off, mutations happen. When DNA replicates a cell, whether it's prokaryotes or eukaryotes, like all organisms will pick up mutations. It's just at random, like the wrong nucleotide will get substituted. However, we do have enzymes that check for those and usually make corrections, but on occasion there are some errors. Not a lot, but enough that over the course of your lifetime they do kind of add up, because if the error persists, then you make a copy of the error, now you have 2 copies of the error, and it'll just keep going. This is called a stochasm, or basically randomness, random chance. So because of this, cells have evolved genes to help combat mutations. So we have what are called proto-oncogenes and also tumor suppressor genes, and the two work in tandem to help make sure that if a cell does pick up mutations that are detrimental to the organism's health and survival, that those cells get eradicated. We do have immune system cells that check for cancerous or precancerous cells if they do manage to make it through the cell cycle, and they'll trigger it to go through apoptosis or programmed cell death, cell suicide, basically. But sometimes, a few cells escape, and it only takes one to slide under the radar to start copying itself, going through cell division, and to make a tumor. So you need to know the names for this group of genes, but you don't have to know a specific gene names. So you do need to know proto-oncogene, but you don't necessarily need to know like BRCA1. Okay? So onco means cancer. So anytime you see onco or hear onco, that means cancer. So like oncology wards or where it's a cancer ward. These genes stimulate a cell to go through cell division. You can think about it like a gas pedal or an accelerator pedal in a car. Okay, they make cell division go. Sometimes a proto-oncogene picks up a mutation, and that turns that gene to be always on. So once it's picked up that mutation, we call it an oncogene. So it's not the DNA that's actually the problem, it's the protein that the DNA makes. So we have a mutation in the enzyme that protein makes, or that DNA makes. So it's always on, and it's always making cyclins, basically. Now you don't have to know these different types of mutations that cause it. Just know that there's multiple ways that this can happen that would stimulate too many of these cyclins being made. Now, on the flip side, we have tumor suppressor genes, which are like a brake pedal. Okay, they make cell division stop. Mutated versions of these are always off. Okay, so imagine if you're driving in your car and you're driving down the side of a mountain, and someone has tied a brick to your gas pedal and also cut your brake lines. Okay, not going to turn out well. Okay, you're going really fast and you also can't stop. So the same thing here. If you get a mutated proto-oncogene so that it's always stimulating cell division, and then a mutated tumor suppressor gene so it's never stopping cell division, then that particular cell is just going to keep going through the cell cycle, and all of its daughter cells are also going to keep going through the cell cycle. So you'll wind up with exponential growth here. Now, cancer, it doesn't just happen with one mutation. It requires several different genes to occur. So we can actually track cancers, and you may have heard, oh, this one's in stage 4, or this one's in stage 2, or whatever. Okay, and that's because it takes a while for these mutations to build up. So there's a cumulative effect, and that's why doctors and the American Medical Association recommend cancer checks at certain ages and certain stages of your life, because if you can catch a cancer while it's still in stage 1 or stage 2, it's really easy to get rid of, and you don't need to worry about excessive chemotherapy. So in this diagram here, this is specific model for colon cancer. So different cancers develop differently, and you can have a different type for each organ and each tissue type that your body has. So that's part of why there is no one cure for cancer, because there's so many different types to it. But in this case, your colon, your large intestine, it's lined with epithelial tissue, which is one of your most rapidly mitosing tissues. It's the same as what makes up your skin. So after the first step, what you would see is something called a polyp. So it's a little growth. We have a little lump of cells that are growing uncontrolled, but they're just staying in one place. Okay, that's after they lose a tumor suppressor gene, and that's the specific gene name is APC. Then if they pick up a second mutation in an oncogene called RAS and a third mutation of a tumor suppressor gene called DCC, then instead of it being benign means neutral, it becomes larger. It's still benign, but we call it an adenoma. So it's in just one spot. So it's a large polyp. But then if those cells lose the tumor suppressor gene P53 and pick up some other mutations, at that point we call it a cancer. It's malignant, mal, bad, mal, evil, okay, same root word, and it's a tumor, and we call it a carcinoma. So carci means cancer, just like onco means cancer. And malignant is when it can start spreading. So again, you don't have to know the specific gene names, but just know, like, it's a series of unfortunate events. And colon cancer is one of those cancers you can't just like look, like you can't, like for breast cancer, you can check yourself for lumps. You can't look inside your own colon. So doctors recommend that you get a colonoscopy after a certain age, which is where, a doctor, they insert a camera and they look to see, are there any of these polyps? Because if you can catch them at this stage or this stage, they could also use a tool to just kind of burn them off and then no problem. But once colon cancer has advanced to this stage, it can be deadly. It's much more difficult to get rid of. So that's why after a certain age, they recommend checks, or if you have a history of this in your family. And we have already seen the case study on Gleevec. Okay. So then there are stages of cancer. So if it's benign, that one's neutral. Malignant is bad. But what's really bad is if the cancer reaches metastasis. Metastasis is the part that's going to kill you. Because if it's not metastasized, that lump, that tumor is just in one spot, and you can have a surgery to remove it. So if in this situation it's breast cancer, you could have a mastectomy, a breast tissue removal, and then that tumor's gone. But if it's metastasized, what that means is little cells flake off of the tumor, get into your blood or your lymph vessels, travel throughout the body, and wherever they fall out, they set up shop and start making new tumors. And cells are tiny, so once it started doing this, you don't know how many cells have reached other places in the body, and you can't just go and cut them out with a surgery. At that point, you need targeted chemo or radiation therapies. So to start with, the tumor will just grow in one spot, but it's eventually going to invade surrounding tissues and stop them from doing whatever their job is. And eventually it will reach the stage where it hits new blood vessels, and it will start recruiting new blood vessels to grow to it. That's called angiogenesis. Angio is for blood vessel and genesis for creation. So it's going to stimulate more blood vessels to go to it, because that's how it gets its nutrients and oxygen. And then once it's reached metastasis, it has all these blood vessels, which are basically a highway system that it can break off into and travel throughout the body. And breast cancer can be particularly bad because the breast is very close to many, many lymph nodes and lymph vessels, which are similar to blood vessels, but they transport different things. So it's basically like, being right near an international airport of your body, so it can go everywhere. So that's why it's important to do self-checks and to see your doctor regularly for checkups. And if any of you have ever read or seen The Fault in Our Stars, which is about cancer, there's a line in that where they say, he lit up like a Christmas tree. So they're talking about someone who's got metastatic cancer, And when they say lit up like a Christmas tree, he was under I think it was an MRI scanner, but it picked up all the little tumors all over the body because it had reached an aggressive stage of metastasis. Okay. And how we treat it, you can have a surgery where you cut it out. If it's in a place where you can cut it out, but you're going to have loss of function. So if it's a, say a tumor growing in your arm and your muscle tissue, if to cut out your muscle, well, then you're gonna, your arm's not gonna work the same after that, but it's, better than dying. But sometimes you can't, like if you get a brain tumor. Now, yes, your neuron cells don't go through cell division, but you have other cells inside your skull that are nervous tissue support cells, all the neuroglia. So when those get cancer growing in your brain, well, some of those tumors are inoperable because you can't cut out certain portions of your brain and still live. So there are other treatments. We're not going to get into them in this curriculum, but there are different medications that focus on stopping cell division. And they've gotten much, much better than they used to be. So now, I told you we were going to go through just a few more items that aren't on these notes, but they are on our exam. So I need to talk to you about causes of cancer. So we've already talked about, I think I spelled this wrong. So just randomness, random chance. So if everyone lived long enough, we would eventually get a cancer, if you live like 200, 300 years, because it's just random. But there are also items called mutagens. There are items called carcinogens. So a mutagen is a thing, some stuff that causes a mutation. Whenever you see gen, that's short for generator. So like antigen is an antibody generator. A mutagen is a mutation generator. So an example of a mutagen could be, like we've discussed earlier, UV radiation, like getting skin cancer from being unprotected in the sun for too long, or nuclear radiation. That's a big one. We do know from what happened with the nuclear bombing of Hiroshima and Nagasaki that those who survived had high doses of nuclear radiation, and that caused many cancers, and most of them were very rare, like rare bladder cancers or rare blood cancers. Your body has to make new white and red blood cells continually, so those tissues are also undergoing mitosis at a high rate. So that's why those pick up cancers more often. I'm going to pause. So you could also get nuclear radiation from other sources. It doesn't just have to be a bomb. So if you look on your class materials page, there's a video I linked about radium, the radium girls. So we used to use radioactive elements to make our watch dials glow in the dark because we didn't know about radioactivity. But we learned about it because a lot of the workers, the women in the factories, they would use a paintbrush to paint on the dials, and they would wet it with their saliva, stick it in their mouth. And so they were receiving repeated doses of radiation in their lips and mouth, and they all developed weird mouth cancers, very rare. Okay, so those things can all cause cancers. We know about those, and we try to avoid them. You do need to know specifically about the Chernobyl nuclear power plant accident. If you have not heard of it, this originally happened in the USSR, the Soviet Union, and they had a nuclear power plant that had a meltdown. Okay? So they were able to evacuate the workers, and they were able to contain part of it, but not all of it. So that whole area is, they've just now opened it to the public where you can go in without a suit on, but it happened in the very early 80s, so it's been like 40 years. But nuclear radiation, if it hits molecules, it then turns those molecules radioactive. So formerly to go in there, you needed, you know, a radiation suit, so that you don't get cancers. And even still, you are still limited to only a certain amount of time, so you don't get a high dose of radiation. But that accident, it was a disaster. It's caused cancers in some of the trees around it, and then the wildlife has had all kinds of freak mutations in it. So it's been a big deal, and you have to basically, when you have a nuclear accident like that, you have to wait it out and wait for the nuclear radiation to decay. It does decay over time, but it takes a while. Now, carcinogens, these are chemicals or molecules that cause mutation, that cause cancers. Cancers. Okay. So carcinogens, the one that you have to know for your exam is smoking, so tobacco. We do know that there is a causal link between tobacco use and cancer. It was hotly contested for many years. And one of the things that was contested was, oh, it's a correlation, not a causation, so you can't be sure. Maybe people are just getting cancer from other sources and not from tobacco. But we definitely know now, we've done enough studies, we've done enough research, it's more than correlational, it is causational. And it does not matter what form of tobacco you consume. So if it's a cigarette or a cigar or chewing tobacco, dip, or vaping, okay, those are carcinogens, you will get cancers. The difference is where you get the cancer. So smoking causes lung cancer. Smoking cigars tends to cause mouth cancer. Dip causes lip and other oral cancers where you have to actually remove your jaw or parts of your jaw. And then vaping also causes lung cancers. And it does so by turning off that P53 gene that we spoke about earlier. There are other carcinogens out there. A lot of items do have labeling on it, especially if it comes out of California. They're very aggressive with their labeling for known carcinogens. There are others as well, like formaldehyde. It used to be used all the time in biology classrooms for preserving organisms for dissection. We no longer use formaldehyde because it is a known carcinogen. And we also take precautions when we have to be around carcinogens, you know, wearing proper PPE to make sure that items aren't inhaled or absorbed through the skin.